the sweeping motion in our body? Certain portion of digestive, okay. Why reproductive? So, female's reproductive system, the fallopian tube or the uterine tube, actually has cilia, and it's there to sweep the egg along. Digestive system doesn't actually have cilia, okay? It has something else because it doesn't sweep the stuff along. Okay. So one of the example is reproductive system in females, the respiratory system, males and females. Okay. Now, flagella is basically a tail right here. It's a tail, and its function is for movement, motility. Okay? Where do you think there is a tail? Sperms. Only sperm. That's the only cell in not female bodies, only males, that contains tail. Okay? Flagella. Then you have microvilli. The function of microvilli is to increase surface area. Okay? So basically, here's a cell. Okay? And on top of the cell, projecting outward, you have microvilli. It increases the surface area. So where do you think you would have it? Digestive system. It increases the surface area for absorption. Increases the surface area for absorption. Okay. Look at my hands. Okay. I have one cell right here, which if food comes, it can only get absorbed right here, right? Now I have a second cell, but it has microvilli. These fingers are microvilli. Now food comes, doesn't the absorption can occur all over here, right? That is the function. It increases the surface area so absorption can. Okay? Does that make sense? Yes.
So do microvilli move at all, or is that only?
No, they don't move.
Okay.
Cilia moves, microvilli doesn't move. It just stays, and there's just area for absorption to occur. Okay, it's like more counter space. Okay. Besides digestive system, where else do you think you would find microvilli? Increased area for absorption. Your urinary system, your kidneys. Okay. Your kidneys have microvilli because it needs to absorb stuff, and you will learn that when we talk about the reproductive urinary system. Any questions so far? Who's ever heard of celiac disease? Okay. Who's ever heard of stuff like, oh, I can't eat gluten? Okay, that gluten-free foods. Basically, gluten-free food is a protein that tends to destroy microvilli. So that is called a celiac disease. So what happens as gluten becomes exposed, somehow in that individual, it basically becomes toxic to the microvilli, and the microvilli are damaged. Therefore, a cell used to be like this, now it's just like this, and absorption can only occur here. Therefore, those patients usually have malabsorption because they can't absorb that much stuff, and basically they have constant diarrhea. Okay? So do you see, if I did not tell you this example, okay, I would have, in the exam, I would have said, celiac disease is in a condition where gluten actually destroys a structure that allows absorption to occur, or increases surface area. Which of the following organelles or structural component of a cell is destroyed? Do you see I did not directly ask you what is the function of microvilli? I kind of explained to you the function, and then from there you are supposed to figure it out. Okay? So things like that. Does that make sense? Okay. Now, these are centrioles. You have two centrioles in our, each cell has two centrioles. The centrioles come into play during mitosis and meiosis or meiosis. Okay? This is the only time doing cell reproduction. Cell reproduction. Okay? This is the only time that these organelles come into play. Their function is to allow mitosis and meiosis to occur. And when we talk about cell replication, I'll get more in detail about it. Huh? Any questions? Okay, ribosomes. composition, it's made up of RNA and protein. Now we have two types of ribosome. We have free or floating ribosome or bound. Okay? Now, this is one ribosome right here. Either it could be bound to something or it could be freely just floating in the cytosol. Okay? Each ribosome, as you can see, it has two different segments, right? One of the segments is called the large subunit. Another one is a small subunit. Okay? So this is the large subunit, this is the small subunit. Okay? Regardless if it's bound or free, it has both segments. Now, when you take the ribosome and you flip it around, okay, so this is the interior part, you can see the large subunit has two different sites. It has a P site and an A site. Okay? A site is for tRNA entry. Okay? And the P site is tRNA exit. So this is where the tRNAs will enter, this is where the tRNAs will leave. Okay? Function of ribosomes, protein synthesis. Okay, function of mitochondria, power plant. Makes energy for cells. This energy is ATP. Okay? So, what is the function of mitochondria? It's a power plant, meaning it makes energy for the cell. What is the energy? It's called ATP, adenosine triphosphate. Okay? Basically, we need money to survive, the cell needs energy. We have cash or dollars where they have ATP. So what is the function? Nutrients, which is stored energy, plus in presence of oxygen, it converts that substance to ATP plus carbon dioxide, and we exhale the carbon dioxide. Okay, so basically from our food, with oxygen, you produce ATP, or the mitochondria does. Now the structure of mitochondria, it has an outer membrane, right here. It has an inner membrane. Between the outer and the inner membrane, you have the intermembrane space. So outer membrane, inner membrane, and then you have the intermembrane space. On the inner membrane, you have a metabolic pathway occurring, which is the electron transport chain. ETC stands for electron transport chain. And then this blue area is called the matrix. Matrix also has a metabolic pathway which is called the Krebs cycle. The Krebs cycle or the citric acid cycle. Okay. Nucleus. Basically, nucleus it contains a genetic material. It's the brain of the cell. It has a nuclear membrane, which is A. Okay? In the nuclear membrane, you have little holes. These holes are called nuclear pores, which is B. What is the function of the nuclear pores? To allow RNA to escape, but DNA to stay inside. Allows RNA to escape, but not DNA. You have nucleolus, which is right in the middle, and the function of a nucleolus is basically a storage site. This is where extra enzymes, extra stuff is stored in. Okay? Now your chromosome, chromatin, DNA, which is our genetic material, is stored in a format called chromatin or chromosomes. Okay? Okay, forget about this part. We're just going to skip this part. Just know that it's stored in a form called chromosome. So C is chromosome, And chromatin is loose structure like this. So loose structure is chromatin, where chromosome looks like a compact structure right here. Okay? Forget about histones and nucleosome. I'm going to skip that part. This is just another photo showing you nucleus and nucleosome. You can cross this slide out because I did not go over histones and nucleosome and anything, so you can forget that. You're not responsible. Okay, ER. Endoplasmic reticulum. It's basically extensively folded membrane system. Okay? You have two types. You have rough and rough ER and smooth. Smooth ER. Why is it rough? Because it has ribosomes attached to it. Okay? It has ribosomes attached to it. Function. Rough ER, protein synthesis. Smooth ER, other macro molecules production, meaning fats, carbohydrates, other stuff, other macromolecules. Okay? So protein is a macromolecule which ER and the ribosomes take its responsibility. Every other macromolecule you need, maybe lipids, maybe carbohydrates, or hormones, they tend to be done by the smooth ER. Okay? If the hormone is protein based, then it's rough ER and ribosomes. Yes. Other macromolecules produced. Okay? I don't know why the whiteboard, as it's getting closer down here, it's losing its, I can't seem to navigate. Up here I'm able to navigate fine. Once the product is made, it sends the products to another organelle called Golgi apparatus. Am I going too fast for you guys? Now, Golgi apparatus function is refines, packages, and ships the material. Meaning, the material that ER just produced, it gives it to Golgi apparatus. Now, Golgi apparatus will refine it, package it, and then ship it. Okay? Example is, let's say your daughter is going to have a birthday party. You're going to have a birthday party for your 5-year-old daughter, and she wants the whole princess theme. That's a Cinderella. Okay? So you go order the cake. The bakers will bake the cake, right? It comes out of the oven. Okay? Do you think they just give you the cake that way? Yes or no?
No.
What happens to the cake? Well, before you package it, you had to decorate it, right? So think of the ER and the ribosomes as baking the cake. Once it's baked, they give it to the decorators. The decorators will basically, put that glass slipper on, the Prince Charming, everything. And then they will put it in a box, okay? And then they will label it, right? This is what Sarah Matthews. So think of Golgi apparatus as the decorators and the packagers, and then they kind of store it for shipment. And ER, the ribosome, are the bakers. How is it able to do its function? Because it has cisterns. Cisterns are, again, membranes, which are folded on top of each other. And they contain enzymes and proteins, which do its function. Yes?
Does the Golgi ship the material to a specific place, or is it just different depending on what?
Oh, depending on where it needs to go. Okay. So what actually happens, it ships it in a sense that material gets put into a vesicle. Mm-hmm. Okay? So its membrane will surround it, and then it will pinch off. And then because it has labels in it, it might go to this cell, it might go over here. Some stuff stays within the cell, some stuff leaves the cell. Okay. But it also labels it. So as you can see right here, stuff is coming in, it gets refined, and when it has to leave, it comes out as little vesicles. And these vesicles will go where it's supposed to. Okay, another just photo. Now let's talk about lysosomes and peroxisomes. Now, I call the lysosomes basically watchdogs and housekeepers. What is the function of a watchdog? To protect the house, right? If there's, let's say, an intruder, they tend to bark their heads off and warn, right? What does a housekeeper do? Clean the house. So that's what a lysosome does. If you have a microbe, okay, virus, bacteria, anything, if it comes inside, so here's a bacterium coming inside the cell, the lysosome will fuse with it. Because lysosome contains basically very, its content is very acidic. So anything that comes in contact with its products basically just degrades. So the bacteria comes into the cell, the lysosome will fuse with it, degrade it, and now it's released outside. Does that make sense? So it acts like a watchdog. It also does the same thing. Let's say the mitochondria is damaged. Okay, do you want a damaged mitochondria to function? No. So what it does, anything that is damaged inside of a cell, it will tend to fuse with it and degrade it. Therefore, clean the house. Okay, clean the cell. Make sense? Okay. So that is the process. It will fuse with the bacterium or the damaged organelle, releases contents, and it basically makes something harmful into harmless. Okay? Now, peroxisomes. I call the peroxisome physicians or doctors. Okay? What is a physician or doctor supposed to do? They're supposed to give you medications to basically detoxify you or basically make you well. So, peroxisomes. Any toxic material which is produced by the cell or you have ingested. Okay? You knew that, okay, today is the first term. So this Friday night, or this Saturday night, you party before you have to sit your, I'm not going to say the word, you have to sit down and study, right? So you went to Yard House, you went to a club, you drank. What do you think helped you or protected you? What part of the cell? Peroxisome. So that alcohol, when it came into the cell, the peroxisome will fuse with it. So alcohol came inside, the peroxisome will fuse with it, and it will make the harmful product into harmless. Okay, something toxic becomes non-toxic. Even a cell, you took a drug, okay, you have infection, you took antibiotic, the byproducts of antibiotic need to be detoxified. It happens via peroxisomes. Okay? So do you see, I could have asked you the same question. You got sick, you went to doctors, they said that you have a throat infection, they gave you antibiotic. The byproducts of the antibiotic had to be detoxified. Which of the following organelles? Does that make sense? You see how I put the same information in clinical case? Huh? Okay, receptors. Protein molecules. Okay, receptors are protein molecules. They're located on the cell membrane, cytosol, or basically nucleus. Okay. Function. It binds chemicals, signals, and causes a change in the interior of the cell, depending on what that chemical is. Either it will cause it to divide, die, transport, etc. So receptor is like basically, think of this door and the lock. Okay? The receptor was the lock, and the key had to be inserted to turn it, and what happened after you opened the door? You guys all came in and class started. Okay? So receptor is like a lock, and the key acts like a signal or whatever that has to bind to it to cause a change to occur. Make sense? Okay, this is a photo of you with all the organelles and little summaries. Okay? Now, I gave you this photo. Work on at home. This is there. I want you to, after you have studied, basically try to see if you could identify these structures and name its function. Okay, we're not going to take time out right now. This is just to test you, so I'm not going to collect it, but see if you can do that. It's a different photo, but again, structures are the same. Okay? Okay, before we start this process, take out a piece of paper. write your name, write if you're 1A or 1B Anatomy, Ontario. So if you are 1A Anatomy, Ontario or 1B Anatomy, Ontario. Always write that.
Doesn't matter where, top right, top left.
No, I don't care. Make sure it's by your name or something. Okay? And so do that and write week 1 participation. Okay? Week 1 participation or week 1 class activity. Did everybody get a chance to sign the roster? Yes? Yes. Let me see the syllabus. It should say. Your syllabus should say. I think 1A, if you have lab tomorrow, it's 1A. If you have lab on Wednesday, you're 1B. Okay. Now, pair up with somebody. You're going to write on your own paper. Quickly pair up with somebody. Decide who you're going to be working with. Okay? Okay. Go ahead and introduce yourself, because you'll be working I want two to a group. If there's an odd number, go ahead. 3. If you have an odd number. So, who are you? So right here, I don't know. Who are you working with?
I work with.
You two, he doesn't have a partner. So you two can, and she can join with him.
I don't have a partner.
1, 2, aren't you? You two, you work with back there. You two, and you two. Okay. You 3, okay. So there's one group that has 3. Now, it's not a test, it's just a class activity. What I want you to do, we talked about all the organelles, right? I want you to start training your brain in a clinical case scenario format. So, 3 organelles, okay? Make a case scenario and a question. So the function or something about it should be asked in a clinical case scenario. Okay? So now you're going to be the teacher making up the question. Okay? If I like any question really well, I might put it on a test. Okay? Does that make sense? So that's why work with somebody. Think of it, it doesn't have to be long. It could be basically 2 sentences. And then make sure you write the answer next underneath it so I know what you were trying to do. Okay? 3 different organs. Or, 1 second. Listen up. It could be 3 different organs, or you could even use something from chapter 1. Does that make sense? Anything that we have learned up to right now, you know, I want 3 different case scenarios. Okay? Make sense? You look confused. You seem confused. Hold on. Is there a question? Yeah, you and your partner are doing the same thing. I just want 2 different papers. Okay? So you work together, make up 3 case scenarios. You write it, and she writes it same exact thing. You had a question? Okay, listen up. Take 15 minutes, and then we will discuss it. Okay? We'll try some students' case scenarios out to see if you guys understand it. Okay? I'm going to go run to the restroom. I'll be back. You can use your notes if you want. Can you close the recording? Okay.